What do we know and what do we need to know about novel coronavirus? Many things about novel coronavirus are spreading rapidly through social media, WhatsApp and the Internet. Some of these could be true, but many of those information are baseless. At a time when the coronavirus epidemic is spreading across the world, it is important to know about some facts related to this deadly virus. Infection: the virus infects the epithelial cells in the throat and lungs. SARS-CoV-2 binds to the ACE2 receptors on human cells, which are often found mostly in throats and lungs. Virus on your skin, lacking ACE2 expression, will be harmless. The virus enters through the binasal passage, eyes and mouth. Our hands are the main instruments that take the virus to reach our mouth, nose and eyes. Washing hands with soap water for 20 seconds as often as possible helps prevent the infection. Infectious dosage: a dose of 700,000 PFU was needed to infect a macaque. PFU, plaque forming unit, is a unit of measurement of sample infectivity. Although the animal did not show any clinical symptoms, the droplets from the nose and the saliva had a viral load. Humans will need a higher dosage than 700,000 PFU to get infected. An animal study on the genetically modified mice with ACE2 receptors showed that it could be infected with SARS with just 240 PFU. In comparison, it required 70,000 PFU of novel coronavirus to get infected. Infectious period: length of time an individual can transmit the infection to others is not known precisely, but possibly up to 10 to 14 days. Artificially reducing the contagious period is a crucial method of reducing overall transmission. Hospitalization, isolation, lockdown and quarantine are all effective methods. Who can infect? Anyone infected with the virus can infect even before the symptoms appear. Most carriers do not even show signs. Covering our mouth and nose when we cough or sneeze will help reduce the infection. The virus is present in the saliva, sputum and feces of the infected person for the whole infectious period. How we infect: transmission is mostly via droplets. This requires relatively close contact, less than 6 feet. This is why it is recommended that we stay 1.5 meters away from each other in public places such as the vegetable market or supermarket. A study done in Hong Kong shows that social distancing can reduce the spread by 44%. Inanimate vector of disease, in particular phones, doorknobs, surfaces are a potential source for transmission, but not much is known about it. It is safe sanitize out hands after touching doorknobs, lift call buttons and counters in public places. How many we infect: the average number of new infections caused by a typical infectious person, that is human transmissibility range R0, is between 2.2 to 3.1. In simple word, one infected individual on the average infects about 2.2 to 3.1 persons. By physical distancing, we can artificially reduce the actual transmissibility, thus slow the rate of infection. Where did the virus come from? It is not from eating bat soup. Once you boil, the virus is decimated. Initially, it was speculated that the SARS-CoV-2 virus jumped from bat to humans. But recent genome studies show first it must have leapt from bat to an intermediary species before it latched onto humans. Another study indicates that a lineage of SARS-CoV-2 virus was circulating in humans before the disease outbreak. How it evolved: SARS-CoV-2 has emerged either by natural selection of virulent strain in a non-human animal host before zoonotic transfer to humans or natural selection of virulent strain in humans following a zoonotic transmission. Only more studies will show which of the two is right. We still are not clear what are the mutations in SARS-CoV-2 that allowed human infection and transmission. When did SARS-CoV-2 emerge? While here have been no documented cases of SARS-CoV-2 before December 2019. However, preliminary genomic analyses suggest that the first human cases of SARS-CoV-2 appeared between mid-October and mid-December 2019. This means there was a period of unrecognized transmission in humans between the primary zoonotic event and the outbreak. Can it infect animals? The molecular modeling suggests that SARS-CoV-2 can affect besides human, bat, civet, monkey and swine cells. Does not infect domestic animals or livestock. Consuming eggs or poultry will not result in SARS-CoV-2 infection. Can one be infected twice? Once we get measles, most of us acquire lifelong immunity. We hardly get measles again. Experimentally infected macaques were not capable of being reinfected. Likewise, there is no evidence of reinfection with SARS-CoV-2 after recovery in humans. However, how long the immunity will last is unknown. How severe is the illness? COVID-19 is not a death sentence. The majority of COVID-19 cases are mild 81%, about 15% need hospitalization and 5% require critical care. That is, the vast majority of the infected will not even need hospitalization. Who are the most vulnerable? Healthcare workers are most susceptible. About 20% of healthcare workers in Lombardy, Italy becoming infected while providing medical care to patients. Among the general public, aged, in particular above 60 years of age and people with prior cardiovascular disease, hypertension, diabetes, and respiratory conditions have a higher risk. What is the cause of the death? Most of the deaths is caused by respiratory failure or respiratory failure combined with heart damage. Leakage of fluid into the lungs, which inhibits respiration and leads to morbidity, is the primary clinical condition. At present, the treatment for COVID-19 is primarily supportive care, including ventilation if necessary. Several therapeutic trials are ongoing, and the results are awaited. Are the virus transmitted by milk sachets, or newspapers? SARS-CoV-2 can persist on plastic and stainless steel surfaces for up to 3 days. When the viral load was 10,000 pfu, it lasted on newspaper and cotton cloth only for 5 minutes. Washing the milk sachets is adequate to remove the virus. Will it spread through the air? In the air, the virus can survive only up to 2.7 hours. Therefore, being in open spaces such as a balcony, the terrace is no harm. Is there a less virulent strain? While many strains are being identified, studies so far have not indicated any mutations that are linked to any changes in transmission or disease severity. Will the onset of summer or rainy season gives respite? No strong evidence exists showing a reduction in transmission with the seasonal increase in temperature and humidity.
To get latest updates in pharmacy, subscribe to Pharma Tutors YouTube channel. Click on bell icon and don't miss our videos.